Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Oh, I can see it in both places. Okay, cool. Right. So I've just flown over from Perth, which is a lot of steps away from here. So it's Perth to Sydney, Sydney to LA, LA to here. So apparently you guys have a 20% speed up on your video recording. So if you want to use that later, that's probably cool. So what I'm going to do is I'm going to talk about the analysis that we're doing on electropherograms and how what we're doing can improve your ability to detect mutations in a sequence when you're just sequencing a regular sample from blood. So you want to detect a mutant variation in sequence. Our aim is to try and make that easy. So just a little bit about who we are and where we come from. Behind that is Perth in Western Australia, which looks a bit like Seattle today with the sunshine and everything and the water. So apparently it rains a lot here. I've never seen it yet, but people say so. The company is basically formed by myself and David Sayer in August 2002. We got completely overwhelmed with the amount of work we had to do, so we hired Corinda as well. Now we're still completely overwhelmed, but we're just better organised. What we wanted to do with the company is we wanted to develop a generic software that you could use for sequencing-based genotyping and that you could use in a clinical situation. The other things we do are, David does molecular biology consultancy. He's got a lot of experience developing assays using DNA. He's also got a lot of experience doing international quality assurance programs, and so we can do that. And the company itself has a very strong collaborative relationship with the Department of Clinical Immunology in Royal Perth, and that's where all the raw data comes from. So David has been a senior scientist at Royal Perth for 12 years, and has been doing sequencing for as long as he could actually do sequencing. So yeah, he's had a lot of experience. He's also had a lot of experience in developing novel strategies, and he developed a lot of the early multiplexing PCR type of assays, where you would try and amplify different alleles of a gene in one test gene. So he's got a lot of publications related to that. This is me, and my career path's been a little bit varied. First, I did laser spectroscopy at Oxford and Arizona State, which obviously very similar places, and no great step from one to the other. Just a bit warmer in Arizona. So I did continuous wave and pulse laser spectroscopy of sort of visible and UV in those places. Then I went to Japan, another very similar place to the other two, and did microwave spectroscopy on a mountain where there was an observatory, a 40-meter radio telescope. After that, I, again following the completely linear line of my career path, went to Singapore and did programming consultancy for telecoms. So I worked for Singtel. And then after that, I moved to Royal Perth, which was the next logical step. The reason I actually went to Royal Perth was my brother was working there as a student, and David, when a student comes into the lab, says, do you know anything about computing? Because we've got all these electropherograms and huge amounts of data, but we're having a bit of a problem analysing them all. And my brother said, no, I don't really know that, but you can ask my brother. So he asked me, and I went to Perth. So Royal Perth has been around since about 1829, at which time it was probably just a couple of tents in the bush. And from there it's progressed a little bit, and there are two sequences in the clinical immunology lab. One is at 3730, which is the highest throughput machine from ABI for sequencing. The other one is at 3100, which is a bit older, but you can knock out 5,000 sequences of 500 to 800 base pairs per week with those. The lab itself does tissue typing, so it types people's immune systems, and the only method that the lab is currently using for that is sequencing. So there are no preliminary methods and no broad typing methods. The hospital also provides a statewide DNA sequencing service, so they'll do commercial sequencing for people outside the hospital. The quality of the data that comes out of the sequencing lab means that RPH is one of the most widely used donor registries in the world for bone marrow and solid organ. And because we have ties with the lab, we've got a good link to novel HLA studies and also a lot of HIV sequence from Professor Morale and his SIBS group. So this is some of the clinical applications of DNA sequencing. The first one is tissue typing, and that's where we've come from. So matching donors and recipients for bone marrow and solid organ. The next one is viral genotyping. So this is the stuff that's being done with HIV. You try and pick apart what components and what genotypes are present in an individual by sampling directly HIV that's been extracted from their blood. You can also do bacterial and fungal genotyping to identify infections, and that's already been done in a clinical case. You can get the identification time down to under 24 hours instead of weeks that it takes to grow these things sometimes. So that's a fairly important clinical application. DNA sequencing can be used to analyze any highly polymorphic gene. Because it's a direct measure of the sequence, it's the most comprehensive way to do those type of studies. You can also do SMPs using DNA sequencing, and as reagent costs and volumes go down, that's becoming more and more commercially applicable. So why use DNA sequencing as opposed to another method? When you do measure a DNA sequence, you identify directly the sequence of bases in that the gene that you're looking at. There's no inferred data. You don't sample a small part of the DNA and then infer what the rest of it is from previous studies. So if there's a novel polymorphism, you always see it. You'll never miss that. It's quantitative, so you can measure the amount of the secondary species or mutant population, and that's quite controversial because most people who've done this previously wouldn't have been able to make it quantitative. You also generate a complete data set, so if you're studying a particular area of a gene and you later find out that there's another nearby important area, if you've sequenced, there's a good chance that you've got that data as well as your original data. So you can, if you do a new analysis, you can probably pick up without having to do more experiments. Also with sequencing, you can use a single platform for looking at clones, looking at single nucleotide polymorphisms. You can score those in populations, and you can do genotyping all in one platform. There's one routine, one set of people, so it's going to be efficient. So this is what happens in the lab, kind of basic overview. You get a sample that comes into the lab, blood or whatever, and you'll extract the DNA from it in one experiment. Then once you've got it extracted, you'll use a PCR to amplify the DNA and select which part of the gene that you're going to actually look at. So once you've got your amplified product, you then do a sequencing reaction to label that so that it can be measured using a genetic sequencer. You do the sequencing and you collect the results. So when you get the specimen, you need to record all the information. So there's a lot of database management that's going to go into making the clinical lab successful. You need to be able to manage all your protocols, and you need to be able to track the outcomes of each stage of each reaction. So that you need to make sure that the extraction is successful. You need to make sure that the amplification is successful. You need to make sure that the sequencing reaction is successful. All of these stages need to be managed in one place. And there's currently no commercial system available to do that easily. You also need to do a full analysis of the electropherograms. And you need to consolidate the results and make sure that what you get out is the same as the samples that you got in and you get the right answers. Also, if you want to do any additional study on the sequence, that's where you get your sequence. So you pick up your ACGTs from there. And what I'm doing is the second to last step, which is analysis of the electropherograms.
Is the PCR amplification
it depends on your cleanup method. If you use a good cleanup method and you've got enough DNA, then it shouldn't be noisy. So if you've optimized your conditions well enough, you should get high specificity, so only the thing that you're looking for and nothing else. And you should get a good amount of that so that the signal to noise ratio is very high. To get good sequence, you're not going to have too much of a problem with high noise. So let's go over what you're doing with sequencing just briefly. So anybody with a fast forward on the video can fast forward that if they know all about it. So first thing you do is you amplify the part of DNA that you want to look at. So the first thing is you get your double strand and you heat it up so that the two strands separate. Then you get a little primer, which is a set of bases that fixes into the DNA at the point you want to amplify. And you put those primers on by cooling the mixture down a little bit so that they only bind to the place you want to bind them. Then you add some extra bases using an enzyme reaction and extend the chains up and get your amplification. And you do this however many times it takes to get a good amount of DNA. So when you've got your DNA, how do you get it into a form where you can measure that sequence of bases? What you do is you do another amplification reaction, but instead of just using regular bases now, you also use some bases that are tagged with a dye. So each dye is a different color, and each of those different colors can be picked up by a CCD camera inside the machine. So if I have a G in my sequence, the complement of that is a C, and I'll tag that with a blue dye, and I'll put that on the end of my sequence, and so I'll know that the 7th base in my sequence is a C when it comes out of the analyzer. And I'll keep doing that until I know what the base is at every position. Okay. Now the number of each of those different length fragments is highly dependent on the rate of incorporation of the dye versus the rate of incorporation of a regular base. So if I have a very good incorporation rate for the dye, then I'll get a good strong signal for that particular fragment length. If I have a bad incorporation rate for the dye, I'll get a much weaker signal at that particular position. So what will happen is, in your electropherogram, you'll get signal strengths that are highly variable, depending on the sequence that's gone before. Okay, so when I get those fragments, I put them in my test tube, and I put them through a capillary, and the rate at which they're drawn through this capillary by an electrical potential is proportional to the length of approximately proportional to the length of the fragment. So what I'll get is I'll get the shortest fragments coming off first, and then longer and longer fragments coming off. And each of those fragments has a label that tells me what the last base is. So the first fragment will come off, and that's got a G label, so it'll go through the laser, and the laser will say, we'll assign on it, and it'll fluoresce at the G wavelength. And the CCD will pick it up and say, this fragment has a G on the end. Next one will come through, that fragment's got an A on the end. Next one will come through, maybe there are two types in there, one's got a T and one's got an A. So that'll give me a mixed base, T and A, for the next position. And you can see in this example that the heights of these peaks are very variable. So it's hard to say, in the case where there's two bases, which base comes from which subspecies. And the variability also makes it more difficult to distinguish between what's real and what's noise, because essentially, if you look at that ab initio, the peak heights are random. So what you measure is a combination of everything that went into that test tube in the PCR, and the peak range is dynamic. So when you measure a lot of sequences, you'll get these profiles that are highly variable, and the signal to noise makes it difficult to pick out some of the heterozygous positions. And you can see that, I mean, this is a nominally a 50-50 mixture, and these ones on the left, they're clearly not showing 50-50 distribution of intensity. The other thing is, when you measure a sequence, you can measure it in the forward direction, extending the sequence, or you can measure it in the reverse direction, extending the sequence. When you do both of those experiments, you find that the profiles are different, because when you're extending in reverse, you're binding onto a different strand of DNA than when you're extending forward. So there's this variation between forward and reverse sequence, which is a help, because if there's noise in one, there won't be likely to be noise in the other.
Why are they different?
Because when you put your enzyme onto the strand of DNA going forwards, behind that, already attached, is a particular structure of DNA. If you put it on the other end, there's a different structure of DNA. So the efficiency of binding at that different structure will be different. So the rate of reaction will be different on the other strand. So the rate of incorporation of dyes will be different. So the profile is different.
Why is the structure different?
So I'm coming in from this side. I've got ACTGGCA, then it binds into the side. Coming in from this side, I've got something completely different, TGAGs. That binds different. You do know which direction you sequence in because depending on what prime you've used to start the sequencing reaction, it tells you which direction you're going. It seems to be a short context. It seems to be a small context because if you make so something like HLA has like a 20% variability between sequences. If you look at 5 bases out from a polymorphism, you look at, say you've got a C or an A, you look 5 bases out the next C, that'll pretty much be the same intensity whether you've got the A 5 bases away. So the context is pretty small. You must be able to analyse those things. And what these peak heights are actually telling you is structural information. And instead of trying to balance the peak heights out using engineering with the enzymes, what you really want to be doing is using that information as structural information to help you do the analysis. Because if it tells you the relationship between two polymorphisms. If I've got 2 sequences that are amplified by slightly different primers, I've got a polymorphism in the primer side. If I then do a sequencing reaction on the products from primer 1 and primer 2, 5 bases down the line, I'm going to have the same profile. So even with different primer sets, once the enzymes move beyond the primer site in its binding, you won't see any more effect from that primer in the profile. For a multiplexing DRB1, I think it's sort of 5 or 6 different ones to cover all the polymorphisms. Sort of 15 bases off. Yeah, have to get David in here. So he's a clinical side. So what you want out of a clinical application is yes or no. You don't want maybe this has got a polymorphism, maybe it doesn't, I'm not sure. So in order to get that yes or no, the best way is going to be if all your peaks are the same height, you know when you've got a homozygous. If you've got a mutant, you want to know instantly that that's a mutation, and you want to, in a 50-50 mix, you want to see half of those mutated sequences going through the sequence and giving you a 50% peak height. But you don't want any background because that makes it harder to do calls, and you want to be able to get the right answer across 1,000 bases, 1,500 bases, whatever you're measuring. So in a clinical situation where you've got a person with 2 versions of each gene, the ability to pick heterozygous is very important. Okay, and people are trying to get towards this regular peak height situation in a number of ways, and one of those ways is manipulating the enzyme itself to make the incorporation of the dye more efficient relative to incorporation of the regular bases. Okay, so guys have done studies on this, and they've discovered that if you introduce certain mutations in the enzyme itself, you can reduce the discrimination against the dyes by large factors. This helps to regulate the sequence height. But even with that, the profiles that I showed you earlier clearly don't have regular sequencing peak heights. So how do we do the analysis of the electrophoregram when it comes off the machine? So first stage, get the data off the machine. Second stage, work out the space of where are the peaks coming, how far apart are they. Third stage, heterozygous base calling, which positions have got a single base, which have got a mixture of bases coming from different subspecies of whatever gene you're looking at. Once you've done that, you want an assessment of the quality. How confident am I that what I called was the right answer? Am I 100% sure? Is it doubtful? Was I 1 in 10 chance of getting it right? How well have I done? Once you've got the bases called, you've now got your sequence of A, C, D, T, whatever, and you need to see how that differs from a reference sequence, or you need to decide which genotype the species that you've measured is from a set. So you do your allele matching to see what you've actually got, if you have a library of previously measured sequences. So, first thing important is the electropherogram. And most of the sequences produce a number of different data outputs for each sequence. First one is what came straight out of the sensor, the numbers that came straight out of the sensor when you measured the diaphorescence from each trace, the A's, the C's, the G's, and the T's. Second one is going to be adjusted, normalized, because your laser power is not going to be the same when you fire that at the dye every time. Temperature is going to vary. The current in the capillary is going to vary. All of these things. So there'll be a second one that's normalized to account for those variations. Each dye will emit light over a spectrum of wavelengths. So if you get an A and a C, there might be some crosstalk between the signals from those two. You need to make sure that you adjust for that. So you make a matrix from some known sequence, and you calculate how much C will come through with an A, and you then adjust your signal to compensate for that. When you pass these strands through an electropherogram, some of them fold up better than others inside the gel. If they fold up really well, they get a little bit less resistance to being pulled through the capillary. So you might see your peak supposed to be here. It might move down here a little bit because it went through a bit quicker. So you might need to adjust your signals to account for that. And it depends what type of dye you put in the end. If you put an A or a C on, you might have a different folding, and you might affect the speed through the capillary. So you need to adjust for those things. And what people have done in the past is they've done a standard sequence, and they've just seen what happens and used that to normalize. And the other thing is it'll just give you a basic intensity information to see whether you amplified anything. So what happens first is when it's been normalized and adjusted a little bit, you get this, which is quite hard to read. So you have to do a little bit more work with it. The way that ABI at least do this is that they divide the sequence up into regions. And each of these regions, they calculate how far C and A have moved relative to T. So if you put a C through, how far has it moved relative to an A in that particular sequence? And you make an adjustment for each trace so that instead of being unsynchronized, like this way. Okay, so I've got A and C, and I might have A's going a little bit quicker so that they do this. So they're out of sync with the A's, or with the C's. So now if I've got a heterozygote, I'm not sure whether this goes with this one or this one. So what I do is I measure some sequence and I make an adjustment and say, I think that I need to compensate the A's by this much. Yeah. So you do a calibration essentially. And that calibration can be done centrally and be applied to many machines, so it's not machine dependent. It's just chemistry dependent. If you use the same dyes, you get the same answer.
Why no competition?
Well, you just pick one of them and compensate everything else against that. So if you pick T as your constant.
I'm measuring A and C in relation to T and delta.
Yeah.
Are you comparing everything against the T?
Yeah.
Okay.
So you just need to decide whether to shift that a little bit forward and backwards. I'm sure there are alternative ways of doing that. You could put it to a function. Because it's the mobility calibration. In fact, the best way to do mobility is to measure the sequence you're going to measure and then use that as a standard. But most people aren't doing that yet. Okay, so you get a raw sequence where the peaks especially somewhere like this, they're not overlapped particularly well. If you do the compensation, then you get a better alignment even though it's not perfect. And that happens because when you've got multiple polymorphisms, they do interact with one another. So if you measure a standard sequence and then you measure another heterozygous sequence, it's still not good enough. So what you want to do is you want to measure the sequence, you're going to measure again and use that. So now you've got a relatively well-spaced set of peaks, all different heights. So you want to do base calling. First thing you're going to do is you're going to decide where you're going to put your limit of detection. So which of these ones are real signals and which ones are noise? And it turns out that's really tough, and that's what limits your ability to detect a mutation. If you've got a high level of background noise and you put a base in there that's actually a valid mutation, but there's only a small amount of that component in your sample when you set it up, you're going to be really hard pressed to pull that out of there and say which is real and which is just noise. So you've got to get above that line, and then you start saying these are heterozygous. There's definitely a mixture here. So there are places in that sequence, and this is a problem where you're not sure. So now you've got to go, well, is that real or is it not real? And one of the ways you can help that is by doing the reverse sequence. Because if that's noise, it won't appear the sequence in the other direction. So that will help you out a bit. So that's one of the main problems. What's your limit of detection? If I amplify two things, if one of them didn't amplify very well, what happens? Do I miss it out? Then my data is not clean. Any further analysis I do is not telling me the whole score. So that's one of the problems with sequence. Okay, so what you do is you say, which bases am I confident I've got right, which ones am I not confident I've got right? And you then make a score that says, for these bases, these are exactly what I want to see. It's a peak, pretty high, it's kind of in a reasonable size compared to the peaks surrounding it. There's not very much background. Its mobility is, it appeared where I wanted to see it. Everything's good. And so most of these peaks here are easy to see. This guy here, that's a C, no question about it. Good quality. This guy, the mobility is not right, so it's more difficult to make a proper call. Is that a Y or is it CT? Have I got an extra base inserted in there or not? So the confidence is going to be less. Same with these two. This case is the signal real. So it's hard for me to say that's a G. So I have to make some note of that. Okay, so it turns out that if you do this analysis over a large number of peaks, you get a good idea of how good your sequencing is. Top sequence, no noise, everything's easy, all the base calls are going to be right, you're not going to have a problem. You're going to get a good overall quality score. As your noise goes up, your quality score should go down, and it should tell you that it's having a harder time doing its job here. So that can allow you to keep your assays optimal. If your assays are optimal, there's no work. You don't have to do any editing. Everything's automatic. Get the answer straight away. If your sequencing is poor quality, it's harder to get the answer. And if you've got a feedback mechanism, you can spot those things and sort them out before it becomes a problem. So what happens if I've got two sequences? I need to score for both of those. Now, if both of those sequences agree and everything's good, I've got an A in both forward sequence and reverse sequence. I've sequenced both directions for this. Everything's good, they agree. Fantastic, good score. This is good. This is harder because this is dragged over from here. So there's little separation between those two peaks. So it miscalls that right. Conflict between the two strands. The overall base score has to be low. So when you do quality control, you need to decide what your main area of quality control is going to be. And when we do genotyping for HLA, we separate the assay into different exons. So we'll amplify one section of the DNA, we'll sequence that, and we'll do a quality control on it. We'll amplify the next section along, we'll sequence that, and we'll do a quality control on it. And if you do that for multiple samples, it's very clear to see which ones can go through the system without any checking, and which ones have got a problem. And this one's clearly got a problem. You can see that the reverse sequence is not resolved properly. It's gone through too quickly, or it's been too concentrated, and the signals have blended together. So this is a piece of cake to call the right answer. This is a really hard to call the right answer. And so what you want to do is you want to be able to see when that happens immediately. And if you plot the core scores across the exons individually, you'll see that exon 3 for this one goes down and the variability goes right up. So it's very easy to see what you need to test. You need to look at these three. These ones are fine. So that reduces your manual workload, because people have been looking at these sequences by hand basically. Go through each base and go, is this an N? Is this an M? Next one, is it a G? Is it a K? Next one. So if you put out 10,000 sequences in a week, and you put out 100,000 bases, that's a bit of legwork you don't want to do. And this is for known genes as well, not volume. Okay, and you can just push that analysis further and further. And if your assay is properly balanced and managed, you'll get a high score every time, and you won't get mistakes, and you won't get miscalls in your electropyrogram sequence. If one of your batches isn't as high quality, the balance is wrong or something up with it, you'll get this huge variability introduced, and everything will go downhill. You have to do a lot more work. So you want to see this the whole time, except for that one. When you design a new assay, you need to know what your optimum conditions are very quickly. Right, so if you vary concentration of a particular component of your reaction, if you want to clean it up with exosat, once you've done your amplification, take everything away that you use as a primer and reagents, you can see what the optimum concentration to use would be to give you the best quality. So if we keep reducing the amount of exosat we use to clean up, the quality will eventually go down to a point where you have to do more work. So, you can then optimize between the amount of work you have to do and the amount of reagent you have to use. And it's always going to be cheap to get more reagent and get something to do that kind of editing. Or you have a job mixing some finance in parts, because they have this different cost of selling. Okay, so once you've got your base calling sorted out, you need to know where to put that against your reference sequence. Where does DNA fit into the gene? Okay, for HLA, what we do is we measure the sequence plus the coding sequence, and the libraries are just coding sequence. So when you do your alignment, you chop off non-coding sequence, and you fit the coding sequence only against the library. So when I've measured this sequence, I've got spillover on both sides of this. It goes into the introns, which is the non-coding sequence. I don't analyze that. I chop it off, and I decide which exon it fits is most consistent with. So once I've done that, it's pretty much aligned. With something like HIV, you don't have that same situation. You just sequence straight through, and you have to align the whole thing, which is a bit harder. Okay, so now I've aligned that, and everything's good, even with that huge dye blob there. So what happens if, against the reference, I have a deletion? There's some bases missing in my sequence. Just need my algorithm to go through and say, hold on a second, I'll just push that sequence over down and get the alignment sorted out. So we put a deletion against reference in there. Okay, now that's fine for a homozygous sequence, no problem. What happens if I've got two different genes in there? I've amplified two alleles together. One's a mutant variation without a polymorphism, one is with. Some guys are doing that separately. They'll do the deletion one separate to the insertion one. So my combined sequence is now got mixed bases in a lot of positions, and it's also got a part of the sequence where there are bases missing in one but not the other, so I have to cater for that as well. The worst situation is when I get two species in the same test tube, one's got a deletion and one doesn't. One, the bases shift up a little bit here, the other one they don't. Now what I get is huge numbers of heterozygous, and how do I pull out part? So that's tough. And in this situation, these are all kind of constant height, and you can't tell which one did this come from strand 1 or strand 2. What about this strand? It's very hard to say. So that's the second problem with sequencing. So what we're doing for HLA is we're matching against the library. So the library may have a certain sequence, and it may not be fully measured your sample comes in here. So you can say for a particular allele how many bases it was fully compatible with and how many it didn't get matched against. Now I've got heterozygous, I've got two contributing species. I can say how many of those bases in my sample were matched against both references, between two references. I can say how many were matched only against one reference and how many were unmatched. And it's slightly complex because I don't know this sequence here. So I've got a S here. S is still compatible with this because it's one of the components of an S as a mixed basis G. But I'm not sure whether that's right or wrong, so I can't call it a mismatch. So it's slightly wrong. For HLA, the library is so comprehensive for the kind of sequencing that we're doing that it's incredibly rare that you wouldn't get a zero mismatch and know the answer. And if you did get a non-zero mismatch, you'd submit it to the database and they'd introduce that as a novel allele. So you'd have to separate those two basically and say, this is novel, nobody's seen it before, we'll submit that and enter it to the database. You can often spot that here. For HIV, it's completely not applicable because you can do anything with any base. So if you see a novel polymorphism, it's no surprise. There's a skeleton. Yeah. There are skeletons there. Yeah. So you've got the skeletal structure of it, but you could imagine a polymorphism being found everywhere. Part of the way that people study HIV, how do they do it? They make a clone and they amplify that clone and they do the structure, they do the DNA sequence of that clone. Right? So what are you sequencing? You're sequencing that clone. What about everything else? You've selectively decided which polymorphisms you're going to see and which ones you're not going to see until you do these mixtures. And when you do the mixtures, how do you know what came from where? So it's very tricky to get alleles out. Yeah. Absolutely. So feedback. Yeah. I'll show you later. Okay. So I observe a sequence from a sample that's got two different strands of DNA in. And that sequence comes out as the first one is an A and a C together. Second one's a C, third one's a T. Down at the end, we've got an A and a G together. How do I know which goes with which? Does the first A go with the second G? Does the first A go with the second A? From that sequence there, because these things are kind of even, there's no way I can know that. So if these are both novel alleles, I don't know the sequence of either of those alleles. I only know that there are two possible combinations. And if there's more ambiguities, then that's going to go up exponentially. So you're going to get a huge problem if you want to work out what the parent sequences are for these things. So this is the third problem with sequencing of a mixture. How can I tell what goes with what? And from that sequence, I just can't. Yeah, so if you had an HIV sequence, you'd be like doing that every couple of bases. Okay, so how can I introduce feedback into that system so that I can get back to I can use the fact that this is the first time I've ever measured an HIV sequence to do it again. Okay, so I've got a list of alleles, I know what they were. Can I make my analysis, I don't improve my analysis because I know that. Okay, and I've got two strands, one's got a K, one's got a T. One of them's got a really bad mobility shift, so I missed the T out, the G out of that second strand. And it looks quite good, so I just called it a T. But I know that the most compatible set of alleles with that particular combination actually would have made a G and a T make more sense. So I put that information back in the base call and say, does one of these strands agree with what I expect? go, yeah, one of the strands does agree with what I expect. The other one's got a slight problem because of the mobility shift. Therefore, I should go back and make my consensus different. And that's the feedback from the knowledge of the allele sequence. But you've got to have two strands to do that, because really you want to have some dispute between the strands in order to get that to be a sensible decision. So that's one way I can use feedback to get improvement in the fitting. It turns out if you do that, because a common problem will be a noise peak. So there'll be a peak that's not actually there, and it'll only be in one strand. And it'll be not actually there in the sense that it comes from something other than the DNA that you're making. So that'll be an artifact of the sequence, but you'll spot that as a polymorphism because it's safer. You don't want to miss a polymorphism. You always want to tell somebody there may be one here to have a look at it, rather than missing it out. Okay, so that'll be very common. But if you go back and you know what the parent allele sequences are, what these genes are likely to be, then you can go back and say that's more likely to be an artifact, so maybe it makes more sense for me to change this first off. And when it does change it, it always flags the quality of that down to 0 to say it messed up the first time. A better way to use feedback is to acknowledge the fact that when you do an HIV sequence from an individual for a particular region of HIV, this isn't the first time you've done an HIV sequence. It may be the 10,000th time you've done it. But the current methods of analysis say, this is the first time we've done this, hey, this is a new sequence, let's check it out. So there's no, people are just not using the huge amounts of data that are available for this. And that's probably a mistake. So one of the first things that you see, if you go in a sequencing lab and you sit down and have to do editing for like more than about 20 minutes, it's really not very interesting. And the other thing is that if you get like these 3 sequences here from different individuals, and they've got the same allele, so the same sequence of bases, these are actually 3 different ones. But you can see here, this pattern of intensities, it's exactly the same every time. It's not changing. This T is higher than this T. T is higher than this T. This higher than this T. These 2 A's are higher than this one. It's the same every time. And if you do that sequencing reaction 1,000 times, you'll see the same thing happen 1,000 times. So it makes sense to say, wait a minute, I've done this 1,000 times before. Let's do something with that. And if you scale the intensity, so I do one reaction, I got a little bit of DNA out, the signal was weak. Do it again next week, got more DNA out, the signal was stronger. So I need to normalize for that. So I scale all the sequences to take account of that. And it's very clear that the intensity pattern is incredibly reproducible from one sequence to the next. So what happens when I use that? I go from the data that looks quite difficult to interpret to data that looks a little bit easier to interpret. So if I've got a 50-50 mixture now, it's incredibly easy for me to see which ones of these contain polymorphisms. Now I don't just gain intensity from this one, I lose intensity from this one. So I've not got one data point now, I've got 2 data points. I know that if I see a polymorphism, I'll have to reduce intensity at second peak. So now I've got 2 places I can get the data from instead of 1. And I also find that these are very regular. There's no problem identifying what the second base is in any situation. So now if I want to automatically analyze that, it's going to be hard for me to make a mistake. As opposed to the previous one, it might be quite easy for some of those heterozygous positions to be mistakenly miscalled. That's easy now. So there's a huge benefit in terms of ability to automate sequencing if you use the fact that you've done this a lot of times before to look at your data. So what's the significance? Problem 1, right? How do I distinguish between background and signal? This is quite small, this is blended. If I do the adjustment, this has gone down, this has gone up. It's clear that this is a heterozygous position, no problem at all. I can call that correctly every time. Same thing here. Probably my map's not so great in that position because of the blending, but still, it's not bad. The next thing is, it turns out, not very clearly on there because of the brightness. Turns out that you could actually quantify these things. So this is not just a mixture of sequenced DNA. I didn't PCR this and then mix the components together. You've got a sample and you just took two alleles, mixed them up, and then PCR'd them. So you get a 80-20% mixture. You PCR that, goes through the whole PCR amplification stages, goes through the sequencing reaction. All of those chemical steps, all of those thousands and millions and huge numbers of interactions, turns out that it goes all the way through the sequencing, gets to the end, and the signal intensity is proportional to what you first put in there. So if you've got 20% of a mutant population, that peak height is going to be 20% of what it would be in a full 100% mixture. And that's incredibly useful and incredibly important, because if you've got disease progression or something like that, if you want to know how that mutant's going, is it going up in population, is it going down in population? If you're giving somebody a vaccine, what's it doing to that population? If you're treating somebody with a particular drug, is that suppressing this mutation or is it enhancing it? So now you can look at a patient, you can go back and you can see what effect that's having on the actual dynamic effect of the population of mutants in that situation. And these guys, Pearce and Statewide, did this work. It's very grateful for them to help me out. This is early data. We haven't been doing this for very long, but it seems to be very reproducible and very easy to get quantitation out. Okay, back to problem 2. So we've got problem 1 is the noise. We were okay with that. Problem 2 is this kind of messy thing here where you can't really see what's going on very easily. What happens when we apply the feedback to this system? Okay, we know what these peak heights are supposed to be. I think it's quite clear which base comes from which strand in that case. You've got one strand that's a minor component, one strand that's a major component, and where the two overlap, you get the bases that are 100% height. There's some stuff that's going to need fixing. You're going to have to fix blending. You're going to have to do some adjustments for things like that. You're going to have to do these adjustments because, when you modify one base, it does have an effect on the next 3 or 4. You're going to have to take all those things into account. So it's going to need a little bit of refinement, but straight away, I can see what 2 sequences I've got there. I can make the computer tell me what 2 sequences I've got there. I can make the computer tell me how that's been affected, how many deletions I've got in one species against the other, and what the actual sequence of both of those species are. So that's a quite big step in terms of analysis of mixtures of samples. So you can get back to directly the components of that system. Provided you've got, if you had a 50-50 mixture, you're going to get in trouble again. So it's actually quite nice to have an unbalanced mixture. So what happens with this ambiguity where I've got several mixtures together, say this Y and this N? How do I know which goes with which? Does this T go with the A? Does the T go with the C? And this is slightly lower in both cases, but it's a bit close. So I'm not sure which goes together. Okay, provided I don't have a 50-50 mixture, it's very easy to see which ones go together. And pull that apart. So now I've got, instead of a heterozygous sequence, but I don't know what comes from where, I've got 2 independent homozygous sequences, and I know what both of the alleles are, all fully sequenced. So what can I do now that I've got that sorted out? I can use sequencing for any type of genotyping. I can make it high throughput because I don't have to do manual work on it. I can knock out with 2 machines 10,000 sequences a week, and those sequences can be analysed on their own. And those sequences can be 800 bases long. I can knock out a high number of bases without a huge number of people needed to support it. You know, if I'm doing a non-clinical or research application, there's probably no need to go and look at that data by hand at all, ever. And because I've got QC, the machine will tell me when I do have a problem or when I do need to look at it. So it'll clear everything that goes through the system and looks good, but it'll tell you when you need to look at something that looks bad. So the amount of labour involved in this is greatly reduced. And this makes it more suitable for the really high throughput systems. If you want to do single nucleotide polymorphisms using sequencing, you can run those through the sequencer much quicker than a 200 base or 800 base reaction. You just run 10 bases off it, take you much less time. So then your sequencer becomes a platform that can do single nucleotide polymorphism screening as well. When you do quantitation, what you can do, you can have a look at, we had a look at a patient with a leukemia, and that leukemia introduced a polymorphism into their immune system genes. We can see that polymorphism, how much. So we can see how it's affected by treatment, we can see if there's relapse, and we can do all that very quickly and quantitatively. So we were able to detect relapses happening before there are symptoms. When you do a transplant, you'll get, with an unrelated donor, you'll get possibility of chimerism, and you can detect when that second type of allele immune system is coming back very early, and you can quantitate that. Okay. You can look at HIV dynamically. You can see how samples are evolving dynamically over time. You take the patient's blood on day one and you take it 2 weeks later, you can see what's happened to the distribution of species in that person, and you can see how your treatment's linking to that. Okay, so you can do novel polymorphisms because your ability to detect polymorphism is enhanced. So what we do, we're able to detect with greater accuracy the presence or absence of polymorphism. These things are fast, so you can get through a lot of sequences in a very short time. You know, if we do 5,000 reactions a week, we can analyse that. Well, 1,000 base sequence, you can analyse that in seconds instead of taking half an hour to look at it by hand. The algorithms are very, very fast and very directed towards library-based typing. So you'll probably know quite a lot about what the sequence should look like before you start. So you can skip a lot of steps in your alignment. You can do it very quickly and very efficiently. Everything's together in the software, you know, so you get your, have a look at the sample, you have a look at the electrophoregram, you have a look at the phenotyping, all of that stuff's all in one place. And with the addition of the peak high analysis, you can move into new applications, you can automate better, and you can do better quantitation. And we've got the software in a lot of HLA labs already. I want to acknowledge these people. Frank and Neil are the guys who are the heads of clinical immunology at Perth. Jody's my brother, and he worked there before I did. Simon and Philip, a lot of the HIV data, all of it comes from those guys. Rebecca and Lisa are kind of like on the front line using it sort of day in, day out. So my aim is to make it better at calling bases than those guys. And everyone else who's doing the sequencing RPH, because they're feeding the data. So that's it. Thanks very much. People are really good. People are really good at looking at patterns. People are really good at saying, this is an artifact. This is a sequence that's dragged through, and they've looked at it 100 times before and they just know. They have a feeling. And you know, part of the thing of doing the software is capturing that feeling and making it into an algorithm. Yeah. So there's the so basically for these typings, you know, if you've done a typing by another method, you'll know what the DNA sequence should have been. You can measure. effectively. And these guys have been allowed for a while. That's very good. The software is, I would say, it's a little bit better, but people will argue with me. And I often have arguments with people who run the labs. The mistrust of software is where it's come from. You know, it hasn't been that good in the past. Yeah. Other people's software. I mean, it's gone obviously through progression. You know, the first time you switch it on, it doesn't go, oh, that's the right answer. Completely got it right. So, you know, it's evolved itself. And, you know, we've got a data set that comes from a particular place, from a particular machine with a particular chemistry. Everything's going to be optimized towards that. If you do something different with it, you know, you're going to have to perhaps think again in some cases. It's surprising how robust it is. If you transfer a platform, you know, if you put it in a different machine, if you use a different polymer to put the DNA through, it's surprising how incredibly robust these things are. And it's just, you know, it's a testament to the fact that these things have evolved. You know, the ones that weren't robust just didn't make it. We're probably, on a single sequence without doing reverse, probably something like, make a mistake, 1 in 500. And it's normally associated with the presence of noise. And some of the time, you'll get a peek through there, and it'll look like a real thing. There's no way you can tell it's not a real thing, but it was just something else in that tube or in that capillary. And there's, you know, what can you do about that? If your sequence is of sufficient quality, you don't make a mistake. HIV is different again. You know, you're much more likely to make a mistake for HIV because you're going to have low-level population. You're going to get the right major species. Or you don't know what the sequence is. You know, there's no gold standard. There's no kind of. We've got the sequence here now. We'll test the software against the known sequence because there isn't one. I'm sure that everybody there is in sequencing are huge because sequencing is the end product of a complex series of steps. You've isolated the DNA from that person's blood. Have you isolated everything that was in there? You've amplified various parts of that. Have you amplified all the species that were in there? Probably not, because your primers are going to bind onto something much better than others. So there's a huge source of, you know, mutations right there. Then you do your sequencing. You know, your efficiency for some of those things might drop them to undetectable. So there you've lost some more. So right all the way till you get to the end, you know, you're not going to get the right answer for HIV the whole time. Yes, and you know, an HIV sequence, everyone might be slightly different, pretty much. So, what you're going to detect is you're going to detect the trends. You know, which ones are surviving the best, and those are going to be the most important ones clinically in any case. You know, the other ones are just kind of like, we'll have a go at this and see if it works, but perhaps not so important. So what you're looking for is you're looking for the ones that are survivors, and you want to know why. But I guess it's directed, yeah, because you amplify what you expect. So there's kind of like a loop there, it's hard to get out of. And you only do that, you try and step through the sequence. You say, we'll step through it a little bit, we'll make a new primer that's in another location, we'll see what that brings about in terms of polymorphism in the last primer site. Can we make a new primer to do that? But it's always kind of bootstrapping upon those things. And you won't get the full answer. But what you will hope to do is get the best answer from that person in terms of what are the best fitted HIV sequences for that person.
So the general belief is that there really is a dominant species that
there will be features of the sequence that will adapt that sequence to be best suited for that individual. And the other thing is, when you're doing a PCR, right, you're splitting this thing up into fragments. How do you know that each of those fragments comes from the same one? It's a tough problem. All this does is it cuts off the ends and it fits them together, so it'll make a consensus out of several different ones, and it'll just align them all together. Because what you'll normally do is when you cut them, you'll cut them so there's an overlap. Software just goes up, which is that. This is a beta one, so if you get your message from Microsoft saying, please contact Microsoft, you can consider yourself contacted. This is beta. This is, I mean, everything's in heavy development associated with this. So I better not browse and do the whole directory, otherwise we'll be there for a bit. To get 10,000 sequences, it's going to be tricky. Just places them. So, you know, you've got this continuous length of DNA, protease and RT here. It just places that in the correct spot for that strand of DNA, and then it looks to find out where that stops matching properly. And after that, it'll cut it off. So the sequence there is tailing off anyway. You've got everything blending together, and it's got too long. But there's probably some insertion or deletion after that, it's just not getting enough handle left of DNA to make that fit properly. So there's that's the end of the whole sequence anyway.
Actually, what I'm saying is you've got your entire sequence and you just cut it out clinically.
Yeah. Yeah.
How controllable is it?
It's completely controllable. So when you've got a PCR, when you're doing your amplification, what you do is you devise two primers. One primer's got a specific sequence that fits into the whole sequence, and the other one's got another specific sequence. And if you design those right, they'll just slot in there in exactly the same place every time and do exactly the same thing every time. So you get the same region amplified every time. So it's completely controllable. And you know, if I use these primers, I always start here and then there. Maximum read length for the machine is kind of 800 odd bases. But you know, you could consider going longer than that if you only wanted to do single-stranded sequence. You know, then you could get like 1500 if you sequence in both directions and you didn't want to have overlap. But mostly you'll do both directions to try and get confirmation of, you know, and improve the signal to noise. RT and protease are friendly. They don't do so many weird things. You get, I mean, that's a pretty regular sequence, not very much polymorphism or anything. This is set up to be so you got 20 mismatches against reference here, so out of, what, 400, 500 bases, so that's a pretty regular sequence. It builds up its map over time. The first time it does something, it doesn't have an idea, so as you keep sequencing, it'll keep building up the map, which is why the last one's better than the first one. and it won't always have information for all the positions. If you've got a novel polymorphism, you don't know what that was supposed to be in terms of intensity. So you'll see some variation in the peak heights even when it's started to get a grip on what's going on.
So when you're using primers to sequence carefully, I assume that you're going after the
They kind of like do judgments on clones and go, in these clones, what are the best places for primer where the variability is the smallest? Yeah. And then sometimes you'll get a sample that won't amplify those primers and you'll have to try something else. And that's when it gets really, really tricky. Or you'll be amplifying two species, one with a deletion, one without, then it's tricky again. So you can probably pull those apart now. This kind of guy here, what was that? Train it up some more and get it going a bit further. That's probably a real polymorphism.
How is the raw data stored? Is it better? So, for example, one of the pieces of data is where what the primers is.
So, at the moment, all of that information is not very well consolidated and is in various places in various databases. And one of the main problems that needs to be tackled is putting all of this together so you get proper feedback. You know, which primers amplify the best, which ones fail, why do they fail, which ones are the best ones to use in the cases when it fails. Now, the software should say, this has failed because you've probably got a polymorphism here, and we found in the past that this primers work well for these guys. And it should tell you that, but you know, those people in the lab, they're doing that by hand at the moment. It takes longer to do those things than it does to do any other part of the experiment. So this is a piece of the puzzle. You know, doing the electrophoregram analysis is a piece of the puzzle. After that comes this huge problem of what does all these species mean? What does this mean in terms of the structure of the envelope? You know, how does this affect the interaction with HLA? And all of those things. Then afterwards, we need to make this easy. We need to make this data go to the people who are doing that easily. So this is the thing we don't have at the moment. These steps are separate. This needs to be fitted better into the rest of the project. Absolutely. Absolutely. Yeah. So all of the raw data needs to go into the database, and you need to add these feedback steps. The better, say you do an analysis and you find that you always see a T at this position for this type of HLA, right? You've measured one and you never saw that T. What do? You just go straight back to the data, and was there any evidence? Was this a bad base call? Did it get a score of less than 20% certainty? Can I pull that information up immediately? Was there a T there and it looked like background? Did it shift? Was there mobility? So all of those things, they should be at your fingertips, and that's why this needs, integration into further systems. These are all different HIV sequences from different people. This is the electrophoregram. But it's yeah. So this is a quality control for that actual data for the single strand. This is for the consensus. We've only got one strand here. It's the same. For the consensus, this is like score from 0 to 50. Consensus would be 0 to 100. If you've got two, and they both give you the same answer, you've got a much better chance of getting it right than if you've got one. So if you put another strand in there, did a reverse one, this will become the same as that. But when I've only got one data point, I'm not going to ever be 100% certain that this is not more. If I did it the other way, maybe it'd be much better chance. For this sequence, it makes no difference. If I had two strands, it wouldn't make a difference. So in this case, it's more complicated because there are 2 strands, but when they agree, you get a good score, and when they don't agree, you get a worse score. And this is kind of my test data for the worst ones. Yeah. Yeah, so that each strand separately has got a score, and then the strands together have got another consensus score. Decision is on top. This is my kind of test set. This is the worst data I have in order to try and make it go on. And that's kind of messy. Reverse selection is much worse than forward. These are all kind of overall qualities of the basis. This is actual sequence. This is actual sequence. This is the reference sequence. So this is a combination of all the known rules for DRV1. This is a reference. This is the consensus of forward and reverse. This is forward and this is reverse. Decision is the yellow one. Yeah.
So on top of the consensus, the second line of letters is the decision, and the last lines of letters.
The decisions for the individual breath.
But it seems that sometimes you don't think decision that expect that you have
These things are.
column, you don't have the same color letters.
Oh yeah, that's only aligned at the point where that bar is. Everything else, these are fixed length letters, and the sequence isn't fixed length. So they're only aligned on that spot where the bar is. You can't see it on here because it's pale. Yeah, the display's only where you're at. Why don't I manipulate the sequence? Basically, you can do what you want, but it's kind of one is time. One is simple, kind of like making every peak width fit 16 pixels or whatever. Can I be bothered to do that? Is it essential for the program? I mean, what we're trying to do at the end of the day is stop people from looking at these things. So, helping them look at it properly. I can compensate. I can make all the peak widths look the same width, but there are other things like mobility shifts. If I adjust the peak width to be 17 pixels and the next one to be 13 pixels and there's one in the middle, it's going to get kind of distorted. Unless I do some kind of smoothing thing between those two, 13 and 17, and compose something like that. So it's just not been a priority, so I haven't done it. It can be done, but just haven't. You can see that these are bad quality, right? They've got red. This one's a good quality one, and this is an easy one. Well, I think I've got it set up to be very, very, very sensitive to mutations at the moment. there's a development one for Statewide, things like that. So this is a kind of work in progress one. I'm trying to do things like, chopping off mobility peaks, making sure that the points of inflection in the right place, all that kind of thing. So it's kind of under variation at the moment, this version. Also, it hasn't got its map yet, so it's untrained. So that's before the nuts axis. The other thing is that if I analyse two different sequencing chemistries, I'll get contamination of my height map. So if I use one type of dye and then I use another type of dye, I'll get variations in these effects. So I have to work all of the dye recognitions and polymer recognitions out in order to make it robust for clinical use. That's pretty good sequence, not very much noise there. Thanks.
Thanks again. You want to take it going now? What we've been doing? I think, first maybe, what's your mental status like? You want to be very technical about it? Yeah, no, we can postpone this till tomorrow if you want.
That's cool. You can slap me around. I'll just stay outside if I don't understand.
Let's do that. Let's give an overview today, then we'll reconvene tomorrow with Simon. Okay? All right.
You were able to actually play things or not able to do that other stuff. Yes.
I can sometimes go a little back. It needs refinement for things like, combining things together, and there needs to be more analysis about how the structures affect the neighbouring bases.
So apparently you're not.
But for that particular one, I could get it out. But I couldn't do it routinely.
The data that that's full marks to the site and when you're actually figuring out depending which knobs you then
Yeah. Yeah.
So here it's whether you actually have to be confident with the certain at certain times of sequence actually get or not.
No. But, I know the way to do it. It's just a matter of, how much time can you actually spend at the computer coding to get that idea into the, into code, Just implementation. There will be systematic problems, but I don't think there's, so what will happen is that it will start doing some sequences first. That sequence that I show you, do that one. and depending on sort of mixture ratios, depending on levels of noise. So you'll get some out first, so you just keep cleaning the data until it's as good as it can be. I'm sure people fight over it. I mean, that data is Simon's data. So, I mean, I guess he's the only one with that kind of data anyway. I think there's anybody else who's got that. Or for mixtures like that. There's plenty of people out there who've got clones. It's got the raw data that's that, every single sequence that's been measured is on there, on the system. Yeah. What the laser was doing at that time,
So I have a meeting for myself. I'll find you. Yeah. Okay. Great. All right, so I'll see you.
All right.
Coffee or something, though?
Coffee would be good, yeah. If I fall asleep, you can slam. Well, check that out. Good.